The overall goal of this procedure is to measure diffusion coefficients of membrane proteins in live cells. This is accomplished by first growing cells expressing the EGFP-Tagged protein and then acquiring a time-lapse image sequence on a spinning disk microscope with focus set to the plasma membrane. The second step is to select a crop in the image stack in which the membrane is flat and there are no moving organelles. Next, the crop is analyzed using case-based image correlation spectroscopy software. The final step is to average the diffusion plots in Excel. Ultimately, live cell microscopy combined with kICS is used to measure the diffusion coefficient of a membrane protein.
This method can help answer key questions in the membrane biophysics field, such as finding the diffusion coefficients of membrane proteins at different conditions.
The first time the kICS code is used, open MATLAB and click File, then Set Path, then click Add with Subfolders and find the folder on the computer in which the kICS code is located. Click OK before saving and closing MATLAB. During acquisition, it is important to keep the membrane in focus throughout the duration of the imaging. Select a rectangular region of interest, or ROI, at the flat part of the membrane where fluorescent